Our next speaker is Daane. He's a cooperative extension specialist based at Berkeley and also Kearney. So I've known, you were the first person I met at UC, I think, when I was working for Fred. So, I mean, for Fresno State, I should say. So anyways, I'll turn it over to Ken, and he'll talk about it more.
Thank you. So what I'm going to do is try to tie in a little bit from Frank's talk to Houston's talk. And I'm not going to end with really solid conclusions in terms of Scaphotopias as a vector, but to open up the idea that we still don't fully understand the epidemiology of this pathogen and what's moving it around. And Frank mentioned the difficulties he's had in doing this work. And you can see up here, I list the postdocs that have gone through my lab working on this project at different times, as different percentages of their work. And so we've had 5 people, including Houston, he's now a professor at UC Riverside, work on this, and it's just been a very difficult vector and insect pathogen relationship to work on. So we heard Frank really focused on Tika as a vector. Houston really focused on Tika in the vineyard. Cindy will later talk a little bit more about how to predict movement of Tika, plowing down, things like that. What I'm going to do today is talk about the different possible insect vectors and why we're opening up the idea that it's not just Tika, but it could be others. And then I'm going to use some recent data from Foundation Plant Services, and I thank the FPS group for their collaboration on this and all the work they've done to kind of compare Tika versus Scaphotopias in terms of, you know, what's explaining the movement at the Russell Ranch setup. So as Houston mentioned, part of what he did as first studies of insects and red blotch was to do surveys of different insects collected in vineyards that had incidences of red blotch. And so we were looking at 5 different vineyards in Napa and Sonoma. And I want to point out that one of the problems with all of these different studies is that we're looking at a point in time and we're looking at a point in space. So I might go out and study 5 vineyards in Sonoma and Napa where there's just not a lot of Tika. And someone else might look at 2 vineyards in Napa next to a riparian zone where there's a lot of Tika, and there doesn't happen to be any western grape leafhopper. So we really need to do broader surveys to get a better understanding of the movement, the epidemiology of this pathogen. But what we showed in this work is that there's a number of different leafhoppers that were positive for the presence of the pathogen that causes red blotch. So the first group, these are all leafhoppers. We really like the Calodonus. Oh, that's not moving down. There we go. The Calodonus and Colodonus, they were in low abundance, but they were about 50% or more positive for a red blotch. So that made them interesting. The Scaphotopias that Houston mentioned, it was high in abundance, and it was about 50% positive for the pathogen that causes red blotch as well. And then what Houston talked about, as well as Frank before, the Tika, membracid, about 15% positive in our study. Now you saw in Frank's work, he was about 28% positive. That's, again, could just be the different vineyards we're studying. It was in relatively low abundance in our study, and in my observations, 35 years working in vineyards, it always has been relatively low in abundance, or at least not very noticeable in terms of being a pest. And then we had a number of other insects surveyed that were in very low percent positive for the pathogen, and they could have been in high abundance or moderate abundance, but we're also interested in looking at these. So one of the first things that I want to mention, we just saw some of these insects that were positive. Being positive with a PCR test does not mean that this insect is necessarily a vector. So with this little cartoon diagram, it's an aphid, we're going to look at the movement of the pathogen. So it could go into the insect, it could be sucked up, go through the gut system, and just be pooped out. So the insect is not a vector. It could go into the insect system, go through the gut process, and work its way into the hemolymph, but it's just in the insect. It's not being taken from that insect into a clean vine. It's not really acting as a vector. What it really should do is be picked up in an infected vine, move through the digestive system, make it into the hemolymph, make it into the salivary glands, and then be put into a clean vine. That's why Frank was talking about dissecting the salivary glands out. So it would just be like if we're going to go out and drink a Pepsi. We're going to have Pepsi in our stomach. That doesn't mean we're going to be able to transmit Pepsi to someone else or something else. So we have to think of these different terms. Frank mentioned this a little bit as well, but just so we're on the same page. There's the acquisition period, the acquisition time. That's how long an insect has to feed on a plant to pick up the pathogen. And that varies in terms of percent efficiency. So you might have an insect feeding on a plant for 1 day, and 10% of those insects now acquire the pathogen. Another insect in that 1 day feeding period, you might have 80% of the insects acquiring the pathogen. There's the latency period. That's that time it takes to move from acquiring the pathogen through the gut system into the hemolymph, and then being able to transmit it into a host plant. And that's what Mark Fuchs did, and we'll talk on that just a little bit later as well. And then you've got the inoculation period. That's also an idea of efficiency. So you might be able to feed on a clean host for 10 minutes, and 1% of those vines are infected. But if you feed for 3 days, you get 80% infectivity. So all three of these things are important considerations in terms of looking at vector efficiency and what really is even a vector. I'm going to kind of, before I jump into some of the examples with red blotch, I started my work with insects as vectors because I was working on mealybugs, and you know mealybugs are a great vector of grape leaf roll associated virus. So when I started this work on red blotch, I thought, this is going to be easy. I've done this before. It was easy with the mealybug situation with grape leaf roll associated virus. We know that pretty much there's a number of different mealybugs that are a vector of this pathogen. And there's a number of soft scales, European fruit licanium scale, that are vectors of leaf roll. It seems like most of these hemipterans that are phloem feeders that feed on the vine are vectors of leaf roll. And in fact, we went out, and this diagram is just looking at different leaf roll strains, except for 2, and different mealybug species. And pretty much any mealybug was able to transmit leaf roll 3, regardless of the mealybug species. So there was no evolution of the pathogen and vector relationship that you see with some systems. We were able to do fairly simple acquisition, latency period, and inoculation trials. This is work with Rodrigo Amida, and we showed that the crawlers of a mealybug were able to acquire the pathogen in less than 1 hour. 1 hour was the smallest amount of time we gave them to feed. They were able to transmit the pathogen in less than 1 hour as well. Now, their efficiency did go up as they had more time to feed on the vine, but at about 24 hours, they started to peak out. We then looked at transmission efficiency, and in the lab, we were getting about 5 to 50% transmission efficiency and about 1 to 30% efficiency in the field. So compare that to all the work that Frank was doing work that I'll show you that we were doing, it's been very difficult for us to get any of these insects to actually transmit red blotch pathogen from an infected vine to a clean vine. In contrast, in field trials, we were taking 10 mealybug crawlers, tiny little insects, and putting them on an infected vine for 24 hours, and then putting them on a clean vine. Those 10 crawlers, I'm sure about 80% of them probably died right off the bat, and yet up to 30% of those vines ended up being shown to be positive for leaf roll. We were able to very easily look at things like, is it semi-persistent or persistent? What this is showing you is we had an inoculation event where the mealybug acquired the pathogen, and then how many days after acquisition was it able to transmit the pathogen to a clean vine? And it showed that after about 3 days, it couldn't transmit anymore. So we were able to show that with each molt of the mealybug, it lost the ability to be a vector. And finally, it was very easy to work with this pathogen in the field because it showed presence of being in the vine fairly uniformly. So what this is showing is that a postdoc in the lab inoculated vines, and you can see the first year there is that solid line on the bottom. Oops, let me go back. First year there is the solid line on the bottom, and the first year we didn't see any symptoms. In the second, third, and fourth year, starting in around July, we started to see symptoms fairly uniformly year after year after year. We know from work that Mark has done, and I think we're going to hear about this later today, it's much more difficult to inoculate a vine with the red blotch pathogen, and sometimes you don't see symptoms for 2, 3, 4 years. Mark Fuchs has said that in some cases, the pathogen from the nursery might actually be in the rootstock, and there you might not see symptoms develop for 3 or 4 years after the vines are planted. So now we'll compare that information with what we're trying to do with red blotch and discovering what vectors are out there. As Frank mentioned, the first paper that identified a potential vector came out of Washington State, and they identified the Virginia creeper leafhopper as the vector of red blotch in Washington. What we wanted to do then was to follow that work up with the common leafhoppers and other common pests in California. We had Virginia creeper leafhopper, but at that time it was mostly in Mendocino area. We really wanted to look at western grape leafhopper, variegated grape leafhopper, and other common insects. So the two that we started with were the vine mealybug, because it's a vector of leaf roll, because it's a phloem feeder, and this pathogen that causes red blotch appears to be phloem limited. And we looked at western grape and variegated grape leafhoppers because they feed exactly like the Virginia creeper leafhopper. We also threw in Tika and Scaphitopius in some of our initial studies because we saw that they were fairly high in terms of the population carrying, via PCR analysis, the pathogen. And we followed the exact same guidelines that they did in the Washington paper. So basically, we had this 48 to 72 hour acquisition period. We then took them from the dirty vine and we put them in a cage with a clean vine where we had 48 to 72 hour inoculation periods. We did this with between 20 and 100 individuals per replicate and 3 to 10 replicates per insect species. So as Frank pointed out, as I'll point out here, in our early studies, we had a maximum of a 6-day period between the acquisition and the inoculation. So that means there was about a 5-day to 6-day latency period in these early studies. So these are the insects we worked with in our round 1 trial. We had the western grape leafhopper, variegated grape leafhopper, Virginia creeper leafhopper, blue-green sharpshooter, tika, a whitefly. Frank mentioned whiteflies were also important to consider. The grape mealybug, the vine mealybug, and leaf-eating form of phylloxera. Results were a number of things. They could acquire the pathogen. And again, as I mentioned, acquisition does not mean it's a vector. Does it transmit? We got a big 0 on all these studies. No transmission in any of this work in the round 1 study. Well, as we were finishing this work up, banging our head against the wall, getting kind of frustrated, Frank's group, as he detailed, showed that tika could be a vector. It's found in commercial vineyards. We know, as both Frank said and as Houston said, and I think as Cindy's going to go through, it does prefer legumes. So this was something we were a little bit curious about because it really didn't like to feed on vines. And in fact, in some studies, it cannot survive from egg to adult on vines. But Frank's group did show that it was able to transmit from vine to vine. So we repeated this work in our study. As mentioned, this was in our initial studies with the 48-hour acquisition and transmission periods. And as I just showed you, we didn't get transmission from this. So we decided to try to change our methodology around a little bit in these round 2 studies. And part of our thinking was that in the laboratory, our source vines didn't have high enough titer for the insects to acquire enough of the pathogen to be good vectors. So what we did in this study is we took the insects to the field. We went to vineyards with really hot levels of red blotch incidence. We caged the insects out in the field, left them for 48 to 72 hours, clipped the cages off, brought that back to the laboratory, and then put those insects onto clean vines. So it was a similar methodology as before, but starting in the vineyard rather than in the lab. So results from this with the Western grape leafhopper, and we did this with Western grape leafhopper, tika, and Scaphitopius. All of the Western grape leafhoppers that we tested were negative for the pathogen. And then, of course, all of the plants that we put them on were negative for the pathogen. So the most important thing for me is that the Western grape leafhopper wasn't even picking up, acquiring the pathogen. If you don't acquire, you can't be a vector. For tika, 13% of the insects tested were positive for the pathogen. And that's very similar to what we saw in the field in our survey. That's very similar to Frank's work, where he found about 19% in the field were positive. In terms of their movement of the pathogen to the host plant, we got all negatives again. For Scaphitopius, 47% of the insects that were put on infected vines in the field were positive. We got all negatives the first year, and we're testing the second year plants again now. So the only positive thing we got out of this was that both Scaphitopius and tika were acquiring the pathogen in the field, making them again strong potential candidates for being vectors. So as this work was being completed, along came the paper that Frank mentioned from Mark Fuchs's group. And the most important thing that I want to point out here, this is looking at 3 different experiments. The end over here is just the total, looking at the pathogen found in the gut of tika, found in the hemolymph of tika, and found in the head or the salivary glands of tika. And again, it's got to get into the head, the salivary glands, for the insect to be able to squirt it into a clean plant. And what Frank mentioned earlier, and what Mark showed in this paper, is that to get into the head, it took a 10-day latency period after the acquisition period. So as Frank mentioned, that means many of our early studies, based really on the work in Washington, had too short of a latency period. And in fact, what these graphs show over here, on the first graph on the left, This is looking at CT values. The lower the value, the more pathogen is in the system. So looking at the gut, the hemolymph, and the head, it goes down in terms of numbers. So the lower the value, the more pathogen is in there. So this is just showing that the amount of pathogen in the insect, in Tika, in the gut, was higher than that made it later on into the head or the salivary glands. And the figure on the right was important because it showed that insects feeding as a first instar, when they developed as an adult, still had the pathogen. So it was transstadial movement. It went from a first instar all the way to the adult. But it goes down in value on the graph, which means it goes up in incidence in the insect. So a third instar feeding had more of the pathogen in it than a first instar feeding because it was losing a little bit of the ability to transmit over time. And finally, this is just looking at transmission studies. Again, looking at 6-day acquisition period. That's when you're starting to be able to see transmission. Again, that's because of the longer latency period. And I want to point out that he did this work. There we go. He did this work both going from beans to alfalfa to beans, beans to alfalfa to vines, and vines to beans to vines. Again, showing you need a long latency period. So what I want to do with the rest of the time then is to focus on Scaphitopias. Frank's group, Mark's group, showed us that Tika can potentially be a vector. Houston showed us that Tika is in the vineyards, but it's not always in very high densities. It can be manipulated through ground covers. We did not find an edge effect. Mark Fuchs found an edge effect. And it didn't always explain the incidence of red blotch that we were seeing in some sites. So we changed our methodology looking at Scaphitopias. There are some reasons we like this as a vector. The most important reason is that this insect likes to feed on vines. From A, B, C, D, E, F, we just see the different stages of Scaphitopias. It feeds on the vines, it lays its eggs on the vines, it causes damage to the vines. And it will even feed, you see there in picture B, it'll feed on the petiole. So it's not always feeding on the leaf mesophyll, which makes it another possible way for it to get into the phloem tissue. What we did in the study is that we took our Scaphitopias, we put them on an infected plant, and we got our Scaphitopias from the field, and we're getting a colony going. We put them on an infected plant, and we gave them a long acquisition period and a long latency period. So after 10 days of feeding, we added a clean plant to the cage, leaving the infected plant in there. Let them feed for a total of 21 days on both, and then took the clean plant out, took the clean plant out and held it for symptoms to develop, and then tested the petioles every 3 months thereafter. Instead of trying to do the PCR work ourselves, we worked with FPS, and they were using a quantitative PCR that develops these CT values based on a fluorescent fluorofluor detection. So these CT values, it's important to note that the higher the value, the less likely the pathogen is being recovered. The lower the value, the more likely the pathogen is there. That's just how many cycles it takes. So anything less than 40 might be a positive. But really anything than less than 30 or down in the 20s, we definitely know it's a positive. So what were our results? When we started this, our dirty plants, our plants that we're using for inoculation, had CT values between about 14 and 24. So clearly these plants from FPS had red blotch inside them. We also found out at this time that our colony of Scaphitopias potentially had red blotch in it already. they had CT values between 31 and 34. Now these were Scaphitopias collected from our vineyards, and our vineyards had red blotch. This is circumstantial evidence, but we are always putting clean plants into our colony with Scaphitopias. And we found out that our colony plants all now have got red blotch. So at the start, we had CT values. Three of the 4 clean plants were not measurable, so they were greater than 40, so they're definitely clean. One had a CT value of 35, which did bring to question a little bit our clean plant stock. The Scaphitopias that we left in these cages for that 3-week period were all pretty strongly positive, with CT values between about 21 to 34. So clearly Scaphitopias is acquiring the pathogen. And our clean plants that we put in there after 3 months, 3 of 4 of the exposed vines are tentatively positive. CT values of 36, 33, and 33. So again, that's in that range which it's probably positive. But we're holding these plants to let the titer increase a little bit to test again to see if this shows up CT values of 16, 20, 25. So it's really strongly positive. So at this point, where are we? We know that Tika is a vector. We know it's been found from various studies to be positive for the pathogen in the field, somewhere between about 15 and 50%. We know Scaphitopias is commonly positive for the pathogen in the field, and this last work I showed you tentatively suggests that it could be a vector. There are things about Tika that are good in terms of maybe it's a vector. Mark Fuchs found that edge effect, which was associated with incidence of red blotch, but it doesn't like to feed on vines, which has always been a little bit perplexing to us. Whereas Scaphitopias, the biology of it makes it seem like it would be a better vector for this pathogen. And I've been doing work with leafhoppers for about 33 years. And I spoke with other entomologists, and we just don't remember seeing Scaphitopias in such high numbers in the 1980s, in the 1990s. And now we're seeing it far more commonly. And we're seeing this increase in red blotch as well. So this is all circumstantial stuff, but that's why we need to continue this work, to get this work done. There's also another whole group of insects that we tested initially, but we tested with that 48-hour acquisition and inoculation periods. And so really we need to go back and retest all these other insects with these longer latency periods. So what I want to do to conclude this in the next 10 minutes is to look at work at FPS with Maher and Vicky and their crew to see if this sheds a little bit of light on any of these insects as possible vectors. And as many people know, the Russell Ranch site has been shut down as a site for their wood, their material, because of the incidence of red blotch which has been found there. So they're keeping this site going as a study site to try to understand epidemiology of red blotch. You can see here the yellow dots show where all these sticky cards have been placed out, both at Russell Ranch and at Classic Foundation. The Classic Ranch has really no or extremely low incidence of red blotch being found. So we worked with them, they're putting the cards out, we're looking at insects, and we're also going out there occasionally and doing sweep net samples. This is just a description of the amount of red blotch being found. And these guys are very carefully monitoring, and they're following up with PCR studies, so we know that this is the pattern of red blotch movement over the years. So 2017, 5 vines, and then an increase in 2018, a bigger increase, huge increase in 2019, 2020 as well. So, and this is just showing it throughout. So we saw this movement over a 5-year period. They also used PCR to describe the different clades of the red blotch pathogen. And that's even more interesting to me looking at vector epidemiology. So you can see clade 1 and clade 2. They're in two different regions of the Russell Ranch block. Also, when you look at it this way, they're going down the rows. So that indicates an insect that's going down the row. If you had ground covers, that would give to me more evidence that it's probably something like Tika that uses those ground covers as a corridor to move down the vineyard rows. But this field has no ground covers. They keep this thing extremely clean. And so then you would assume if Tika was a major player, there might be a little bit more of an incidence near that alfalfa field at the top of the graph. But we really see the incidence spread from within the vineyard itself. And that might lead a little bit more to thinking, well, it's an insect like Scaphitopius or a mealybug that's inside the vineyard feeding on vines. So if we look at the density, both in Classic and Russell Ranch, and the other one is just the CAC Woody Index vineyard. So looking at Classical and Russell Ranch, they both had very low incidence of Tika in the top graph. And in the bottom graph, they both had higher incidence of Scaphitopius, but more Scaphitopius in the Classic than the Russell Ranch site. If we look at the insects that are really in high abundance, well, it was the western grape leafhopper, the variegated grape leafhopper, not much in terms of Virginia creeper leafhopper. So these things were at 10 to 20-fold higher incidence than even the Scaphitopius. And we know that these things are just not good suspects as vectors. But they were there in high incidence in both Russell and Classic. And then we had a number of these other insects that tested positive for the pathogen in our earlier studies. The Ceciid especially is kind of interesting to us, the Calodonus and Colodonus, and even Lygus, although we don't suspect Lygus as being a vector. So many of these were found in both Russell Ranch and Classic blocks. So we're looking here right now at levels of Tika between the Classic and Russell Ranch blocks. And what we show here is that the incidence in both was really pretty much the same. Minor variation between the different years, but pretty much the same incidence. And just as Houston mentioned, as I think Cindy is going to talk about, incidence really was peaking towards the end of the season as they're moving in from someplace else, or in some situations in some vineyards as the ground covers are dying down. So what does this mean in terms of control? Well, the Russell Ranch site was sprayed with more insecticides than you can legally put on. They sprayed all the time trying to get a handle on this. They were not roguing because they were looking at this as a study site. We had just as much Tika, almost as much Scaphitopius in the Classic Ranch site. And they had almost no spread of red blotch during this same period of time. So the first thing that this says to me, regardless if it's Scaphitopius or Tika, you're not going to spray your way out of this problem. It also says to me that roguing is extremely important because that was the main difference. All the different insects were, slight differences between insect species between Classic and Russell, but the most important difference is that Classic was roguing all the infected vines out immediately, and Russell was leaving them there. So in summary, we've done a number of field surveys and transmission tests. Western grape leafhopper, if that work in Washington were correct, would probably be our main suspect because it feeds very similarly to the Virginia creeper leafhopper. But in most of our field surveys, it just is not very high in incidence in terms of having the pathogen. And with the Western grape leafhopper and the vine mealybug, We did do very long latency period tests, and we never got transmission. So I just don't think the western grape leafhopper or the mealybugs are vectors. Tika has been shown to be a vector. We are finding it on the vines. It doesn't prefer vines, but it is a known vector. So we do know that this insect is moving the pathogen around. We still have to look at ways to controlling it to try to reduce incidence. We can't say for sure that Scaphitopius is a vector, but we're leaning that way. And if so, we have to develop control programs for it. But probably from what we saw at the FPS site, it's not going to be spraying as much as it's going to be roguing. Houston talked about cultural controls, mow and discs. That was focused on Tika. That is one way you can reduce the incidence of that insect in your sites. And again, looking at FPS, the take-home message for me from FPS is that roguing is critical and that you're not going to spray your way out of this as a problem as long as you've got fairly high incidence in your vineyard. So with that, I thank you, and I'm at 10:40, so any time for questions, I'll take them.
We have questions. There are questions from online.
Okay.
So I have a couple questions from online. One is, if red blotch can stay dormant in nursery plants for years, are there viral tests that can detect the virus while it is dormant, particularly in dormant nursery materials prior to being delivered?
Yeah, hopefully one of the viticulturists can address that later on. So what I'll do is I'll tell you what Mark Fuchs, a virologist, suggested to me. He thought that if it's in the rootstock material, you'd have to test the rootstock for it. And it could be in very, very low titer. And the reason it might be taking 3 years or 4 years to show symptoms is because of the time it's taking to go from the rootstock into the cyan and then expressing itself by getting higher titer.
There's another question. What is Scaffy distribution in hillside versus valley floor vineyards?
So we've been finding Scaphitopius now in the Central Valley. We've been finding it in the Central Coast. We've been finding it in the Sierra foothill region, and we've been finding it in Napa and Sonoma. And again, that's something that maybe I missed it because I was young, and we're seeing it now because we're more interested in it. But I spent the mid-'80s working on the variegated grape leafhopper, and I can't believe that I missed this being in the densities I'm seeing now. And we had sticky cards out there, and so maybe there's been a change in its density. These are all unknowns. I wish we had more answers by this point in time.
One more question just came in. It would appear that prevailing winds may play some role in spreading. Is this correct?
That I don't know. I would say more, and I hope Cindy addresses this. Houston touched on it. I would think for Tika, it definitely would be proximity to legumes, and that they're going to move out from there. With Scaphitopius, it seems to be spread throughout the vineyards fairly evenly. And again, there's these other possible vectors that now we know we've got to retest because of the latency period.
I have one question for Houston that came in after he had finished. And it's, could Scaffy populations be controlled while they are eggs before hatching?
We just don't know. I mean, that insect is hardly studied at all. It's just coming on our radar. Like Kim was saying, it's something we've kind of seen peripherally, but it's, potentially, but we would have to look more into control strategies. It's similar to Tika when we first identified that as a vector, and we knew it was in vineyards, and there's all this control information in these annual cropping systems, but there was nothing we could immediately transfer over to vineyards in terms of chemical control. So I'd say the similar case with Scaphitopius. We don't have chemical control recommendations for it at this point. We don't even know if you need to do that, I guess. Right.
Thank you.
Okay, well, this brings us to our first break. So we will take a break till 11 a.m. Let's have some coffee and a carb up before we come back. So I'll see you back here at 11 a.m. sharp. Thank you.